inotropic eye, when it's flattened. That's what we call inotropic eye. Normal resting condition of the lens is when you have it flattened. So that is relaxation of your ciliary muscles, right? Far point vision, but when you're looking far, more than 20 feet far, you look at point at which the lens does not have to thicken for focusing to occur, normally 20 feet or more from the eye. So that's when the lens changes the shape. Near point vision, closer to 20, closest point, an object can come to the eye and still be focused. Actually, if you try to bring your finger, you'll see there's a point when it's really blurry and you can't focus it anymore, right, really close to it. So there's a point where you can, keep it focused very close, but after that, you can't. So this is again showing the picture of your distant vision and near vision here with your lenses and focal points. You can see the area of the focal points here in distant vision is smaller and your near vision is bigger back here. So this is when your lens is flattened in your distant vision, but when you're coming nearer, the lens gets thickened and starts getting suspensory ligaments starts pulling it. Functions of the complete eye again. So when an object is less than 20 feet from the eye, the image falling on the retina is no longer in focus. So 3 events must occur to bring the image into focus. You have again accommodation. We looked at accommodation for smell, right, and how that works. So we have accommodation by the eye too. Constriction of the pupil and convergence of the eyes. Accommodation by the lens, this is contraction of ciliary muscles causes the lens to become more spherical. Change in lens shape enables the eye to focus on objects that are less than 20 feet away. Constriction of the pupil is now increasing the depth of focus. So that is when you constrict the pupil. Convergence of the eye's medial rotation of the eye. So these 3 events must occur to bring the image into focus. Accommodation, constriction, and convergence. Structure and function of the retina. Now, retina is, this area here of the, which layer is it? The retina. It's the nervous layer, right? Where you have the nerve cells? Nervous layer. So there you go. So you have all this pigmented layer here where you have the pigmented cells. And then you have all these nerves, right? You have the photoreceptor cells and then bipolar nerves connecting. You have interneurons and all these other kinds of neurons that connects in this area. And then you have the ganglionic area, and that brings it to your optic nerve and sends it to your brain. Where in the brain does it send it to? Vision? You know this. Vision. Occipital, right? No. Pigmented layer of the retina provides a black backdrop for increasing visual acuity. Rods and cones synapse with bipolar cells. So you have rods and cones that synapse here. Bipolar cells synapse with ganglion cells which form the optic nerve. So here, when you zoom into this area of your retina, this is what you see. You have the choroid out there, and this is where you start your pigmented layer with the pigment cells. You have all your photoreceptor cells in here. Cones are the red ones, rods are the blue ones. So you have rods and cones, the photoreceptor cells, connecting to your bipolar layer here of neurons or bipolar cells. And you have some other cells that connect them in between too. You have the horizontal cells that go this way. You have the amacrine cells that goes, button. And then you have one connecting here, interplexiform cells. Those are in between connecting your bipolar cells. Down here you have the ganglion cells in your ganglionic layer. This cells is what you have the cell body that you have the long axon that goes to your optic nerve, because your optic nerve have the axons, right? That brings it to your brain area. Direction of action potential propagation here. So you start from here. This is where your photoreceptors capture it, right? And depolarizes starts from here. So action potential travels all the way here, through this to your ganglionic layer, optic nerve to the brain. And this is the direction of light that light comes in from. So this is where light comes in right here. Now let's look at the structure of rods and cones. Rods, so we know rods are what? What are rods and cones? For night vision? Yeah, but what are they? Rods and cones. They're cells, right? What kind of cells can they be? Photoreceptor cells, right? That is your photoreceptor cells where you're catching that thing. So they're responsible for non-color vision, black and white vision we say, and vision in low illumination at night, so nothing colored. Anything got to do with colors is your cones. So rods is night vision. Rod-shaped photoreceptive part of the rods contains about 7,000 double-layered membranous discs. 7,000. Discs contain rhodopsin. So this is a protein that they contain a purple pigment consisting of the protein opsin, covalently bound to a yellow photosensitive pigment called retinol that we get from vitamin A, is your rhodopsin. So your rods contains a lot of these rhodopsins in there. So this is how they look like. You have this rod, this long blue one. You have this outer segment that have discs in there. These discs are just folding of the outer membranes in there. When you zoom into the disc, you'll see this in there. You'll see the membrane, right? The double membrane, phospholipid layer, and you'll see the outside disc. Inside, you have this opsin protein, like a receptor molecule here, and you have a retinol that we get from vitamin A inside the opsin there. So when the light hits, right, the light depolarizes or starts this action potential, and it starts from here. The retinol is the one that changes shape first, but we'll look at that later. But this is how this looks like. You have the outer segment, inner segment, you have the nucleus, the soma, the body area, you have the terminal of the neuron, which we call the synaptic ending. Same thing with the cone. So here the cone is a little smaller. Exposure to light activates rhodopsin. Rhodopsin is split by light into retinol and opsin. So you saw how you had the retinol in here, right? You had the retinol inside an opsin there. So when the light hits, first thing that happens is you have it activated and they split. Retinol and opsin, they split. Eventually that results in an action potential by, you again have the G proteins and all the ions coming in, opening. So eventually that gives action potential. Light adaptation is caused by a reduction of rhodopsin. So that's when you get light adaptation. That's why when you first go into from a dark room to a light space, right, you get you're trying to adapt, you're blinking your eye, you're it's so bright. But after a while, you get adapted. Same thing happens when you go from light to really dark. You get a adaptation period there as well. That's not as dramatic as light. So this is what you should, know from this area here, the photoreceptors. When you have the light coming in and hitting this, you have your rhodopsin activated, right? So you can see how the retinol is inside the opsin. And now in step 2, here, it's the light activated rhodopsin, and you can see from purple it became white.